Hello everyone, welcome to today's webinar. I'm Alexander Taylor, contributing editor at CNEN, and I will be moderating today's event. The webinar is titled Small but Bright, µSEC-MALS adds light scattering to UHP-SEC for biophysical characterization of biotherapeutics, and is being sponsored by Wyatt Technology. CNEN works with sponsors to identify topics of interest and value to CNEN's audience, and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNEN online after the live webcast. The presentation today is being sponsored by Wyatt Technology. Wyatt Technology markets itself as a recognized leader in light scattering instrumentation and software for determining absolute molar size, mass, conformation, charge, and interactions of macromolecules and nanoparticles in solution. During the presentation, we will hear from Dr. Stephen Koza, a Principal Applications Chemist with Waters Corporation, Michelle Chen, Director of Analytical Services for Wyatt Technology, and Mark Havrick, a Senior Scientist in Sterile Product and Analytical Development with Merck. I will now hand over to Stephen.
Thank you, and thank you for joining our webinar. I'm going to start this webinar off by just really talking about the basics of SEC and where small particle technology plays a role in SEC separations. On this slide, what we're going to talk about are the principles of size exclusion separations. These separations are really based on the size of these proteins in solution. And ultimately, as we have a packed column, the pore size and pore volume are really the critical parameters that we're dealing with. And if we think about this separation as shown on the bottom of the slide, what we have are pores of different diameters and different sizes. If we have a larger particle, that particle can actually explore lower amount of the pore volume that's available on the particle, so it spends less time in the pore, more time in the mobile phase, so it comes out early, as shown by the dimer, the red dot. A smaller particle can actually explore more of the pore volume on a particle, so it spends less time in the flow, so it comes out later, as shown by the blue dot or the monomer. The important things to remember in these separations is that ideally, these separations are isocratic and there's no absorption of the protein to the surface of the particle. What that means is the K prime is essentially 0. Because of that, this is a very low resolution technique. All of the analytes are going to elute in a column volume of less than or equal to 1. If we compare that to other separations such as reverse phase or ion exchange, where we can run these extensive gradients, we can actually have analytes elute in column volumes much, much greater than 1. Shown in this slide is a picture of a particle. It's a 1.7 micron BEH particle, and you can see on the left that the entire particle, if we're looking at the surface of it, it's a porous structure, and this is what we're allowing the proteins and peptides to elute in and out of, or basically diffuse in and out of. If we take that particle and with a broad ion beam we cut it in half, we'll get an image like we see on the right. This is the interior of the particle. You can see that this pore structure extends deep within the particle. So ultimately, our proteins and peptides are going to migrate inside of this particle and eventually diffuse back out. So you can tell that by having a smaller and smaller particle, that the amount of time that's going to take just from random diffusion, random walk model, it's going to take less time, so we're going to have faster separations as we go to smaller particles. We'll show you some data about that later. So briefly, I'm going to touch upon column selection and method development as you start with a protein and you have to develop an SEC separation. There are generally three types of particles that are used for SEC separations. The original and also still commonly used particle, as shown at the top, is the cross-link agarose type of particle. These are gel particles. They have very poor mechanical strength. They're kind of squishy, but ultimately these particles are somewhat advantageous in the sense that of these three different types of particles, they have the lowest levels of secondary protein interactions, and they also have really good basic pH stability. A number of years ago, silica particles were introduced for SEC separations. The huge advantage of silica particles is they can be made smaller, they have good mechanical properties such as strength and pore volume for SEC separations, so they can basically take run times and make them much shorter for these separations. Silica particles have limitations in terms of the fact that they have higher residual silanol activities, or very high residual silanol activities, even though we try to cover those with some type of ligand such as diol. The other disadvantage of silica is that it has limited stability at basic pHs. Somewhere in between these two particles in terms of secondary interactions and basic pH stability are the water's BEH bridged ethyl hybrid particle. These particles have equivalent mechanical properties to silica in terms of strength and pore volume, but they have reduced silanol activity and significantly higher basic pH stability. In general, we're going to basically pick a SEC column based on the pore size that we have. And the pore size is really going to be dictated by what we have for the size of our proteins. And you can see from these electron micrographs of some BEH UPLC particles, as we go from 125 angstrom pore size to 450 angstrom pore size, you can visibly see that these pores are actually enlarging significantly as we go from left to right. So one thing that vendors, manufacturers will do is generate these calibration curves using protein molecular weight standards. Now remember that this is a size-based separation, not a molecular weight-based separation, but we kind of throw this out as just a general rule of thumb in terms of kind of looking at proteins of about the same molecular weight and figuring out where they fit on these calibration curves. Ultimately, you can use this to look at the proteins that you need to separate and look at these calibration curves. And what you want to pick is an area on these individual calibration curves where you more or less have a pseudo-linear region. And that will allow you to get the maximum separation out of the SEC separation that you're trying to get. If you have a wide variety or a wide range of molecular weights in your sample, one thing that can be done for proteins, much as it's been done for years for polymer analysis, is combine proteins or columns with different pore sizes. On the top chromatogram, we have a standard 200 angstrom pore size column, and on the bottom we have a 450. You can see that the resolution between the larger species 1 and 2 isn't as good in the top chromatogram, but better for the separation between peaks 3 and 4, which are smaller proteins. If we look at the bottom chromatogram, peaks 1 and 2 are much better resolved, but peaks 3 and 4 are not as well resolved. If we look at the middle chromatogram, which is basically the same total length as the upper and lower chromatograms, but a mixture of the two pore sizes, we see that we kind of get a adequate separation between peaks 1 and 2, which is about 1.3 kilodalton thyroglobulin dimer versus a 700 kilodalton thyroglobulin molecule, and peaks 3 and 4, which are IgG and BSA. So if we have a wide range of molecular weights to separate, we can combine columns of different porosity or pore size. Particle size is another important factor. On the left pair of chromatograms, in the blue, we see a 5-micron silica-based SEC separation. On the bottom in black, we see a 3.5-micron SEC separation. And you can see just by going from a 5-micron to a 3.5-micron separation that the peaks are sharper, more well-resolved. We have higher efficiency for this separation. In a more extreme example, if we look on the right, for 450 angstrom columns on the top, we have an 8-micron silica-based separation. On the bottom, we have a 3.5-micron BEH-SEC separation, and you can significantly see the improvement in resolution as we change to smaller and smaller particle size. This carries on even further as we go to sub-2 micron particles. On the top separation, we have our previously seen 3.5 micron 200 angstrom X-bridge column, and what we've done here is run two of these in series in order to get the same plate number as we have on the bottom separation, which is our UPLC 1.7 micron particle column. So ultimately, you can see that we get very comparable resolution between these two separations. The difference is that the column length is much longer on the top and the linear velocity is much lower, such that we can get the same resolution on the bottom in 7.5 minutes that we got in 37 minutes on the top. So ultimately, the advantage of going to smaller and smaller particle sizes for SEC separations is that we can increase our sample throughput. And this is really driven by the Van Deemter curve. We at Waters love Van Deemter curves. And shown on the right is a Van Deemter curve for small molecules, which diffuse quite readily. And what we have are three terms, our A term, our B term, and our C term. The B term is our longitudinal diffusion term, so how much the particle or the analyte diffuses back and forth in the column, and that basically leads to a broader band and loss of resolution between two peaks. And you can see that that has a significant effect as we get to really lower flow rates. In general, for proteins, since they really don't diffuse, it's really hard to find these B terms, as shown in the bigger Van Deemter plot, shown marked with the 4 micron and the 1.7 micron labels. These are actual Van Deemter curves for an IgG on these two particular SEC columns. The more important thing to notice here is that the A term is significantly lower, the A term being the eddy diffusion that occurs between the particles. Since these particles are small, the eddy diffusion term is lower, and the C term, which is basically the transfer in and out of the particle, or diffusion in and out of the particle, which we can imagine as the smaller the particle is, the more rapid that transport is, or the diffusion is in and out of the particle. So what we can see is that ultimately, as we look on the Van Deemter, if we compare the plate height, and if you remember back to your introductory classes in chromatography, the higher the plate height, the less resolution we're going to have. Basically, the lower number of plates we're going to have per column. So the lower the plate height, the higher the efficiency, and the smaller the particle size, the higher the efficiency we're going to have in our separation. And we saw that from those previous data. Another important part of these separations is to really deal with the chemistry. So this is an example using an AutoBlend Plus feature on our H-class bio, which basically takes concentrates of phosphate buffer and salt and makes different pHs from pH 6 to 7.5 and salt concentrations from 150 to 350 millimolar sodium chloride. And you can see that as you go to high pH, low ionic strength conditions, that this SEC separation for an IgG really kind of degrades. We see a loss of resolution, and we see basically a loss of recovery of the high molecular weight species. As we go to the higher concentrations of salt and lower pHs, we see an area where the thumbs up is, where we basically see, even though we have changes in pH and modest changes in ionic strength, that this separation is very reliable and reproducible. So we want to, as we develop these methods, find conditions in which we, with small changes, we don't see significant changes in our separation. So now we're going to talk about the systems that are needed for these separations. As we get to smaller and smaller particles, we wind up having to go to smaller and smaller IDs of columns, just from a manufacturability standpoint. So we need systems that can deal with this. Remember, this is an isocratic, non-binding K prime equals 0 separation. When we talk about extra column dispersion in a system, what we're talking about, if you can imagine, as shown on the bottom, is we're injecting fundamentally a zero volume injection of some analyte. And what we're doing is sending it through the system without a column, through the autosampler, a union, and a detector, and we're measuring the volume of the peak that comes out. If the volume of that peak that is significant relative to the volume of our SEC peak, it will basically reduce the resolution of our separation. And I will show you on the next slide an example of kind of how we can think about this. In this slide, what we have on the top set going across is our pre-column dispersion, our dispersion on the column, and our post-column dispersion. If we run a UPLC system with a 4.6 millimeter ID column, we're going to wind up with a injection where we lose a little bit of resolution just from the injector into the head of the column. So if we could imagine that peak before it gets to the column in the upper left, we can see that the peak is still fairly sharp, but it's broadened out a little. It's not zero volume anymore. That peak disperses even more in the column, but that's okay. We want that to happen. That's how we drive our separation. And then as it leaves the column and goes into the detector, it'll disperse even more, and we'll lose some of that resolution. By contrast, if we do that same experiment on a higher dispersion system, shown in the middle chromatograms going across, where we're looking at a HPLC column with higher dispersion volume, if we inject this narrow peak, we'll wind up with more dispersion of that peak before it even gets to the column. And then because these peak volumes are much smaller coming out of the column, if we imagine this peak right as it comes out of the column, the resolution will already be degraded relative to what it would have been on a low dispersion injector system. And then when we take that peak and further translate it into a detector that's more high dispersive, we'll even lose even more of that resolution. So even though the column's the same, because the system dispersion is higher, we'll have significantly lower resolution for our separation. The way to combat this is to use a higher column ID and a higher flow rate so that the peak volume becomes significantly higher, as shown on the bottom chromatogram, but because the flow rate is higher in the time domain, the peaks look just as sharp, and we wind up with the same resolution as we did on the top. And this is an example where we've taken a UPLC column, and on the top we run it on a low dispersion UPLC system, and the resolution between the two principal components is 2.37. If we take that same column, same flow rate, same conditions, and run it on a higher dispersion system, an Alliance HPLC system, we can see that we lose a significant amount of that dispersion. So if we're going to go into smaller particle IDs for UPLC SEC separations, we're going to have smaller ID columns. We need to have systems that have very low dispersion throughout, from the injector all the way through to the detector. And if we look at a typical vendor, and this is our Waters information for LC categories from HPLC, UHPLC to UPLC, we have recommended column IDs for these. This is with K primes that are significantly greater than 0. So if we consider an SEC column, what we would recommend is, as you go from HPLC to even UHPLC technologies, that the larger column IDs are always going to give you better resolution than the smaller column IDs. And pushing 4.6 millimeter into an HPLC or UHPLC separation will give you a loss of ultimate resolution. So you really need lower dispersion systems. So we need 4.6 millimeter ID columns for UPLC SEC separations. So as a couple of hints as we kind of finish up my portion of the separation, we're just going to talk about some pragmatic issues in the lab. When running these separations, it's important to remember, because this K prime is 0, there's no pre-concentration at the head of the column, that the critical components include the autosampler, any dispersion that occurs in the tubing and connections between the autosampler and the column. These are critical. If these peaks broaden at this point, they'll never get narrower again, much unlike they would in reverse phase separations. On the other side, much like any other chromatography separation, we are concerned with the dispersion in the detector, as well as the connectors that we use between the detector and the quality of the connectors between the detector and the outlet of the column. So it's important to set these experiments up correctly and to make sure that our connections basically aren't adding any extra column dispersion to our separations to get reproducible results. And another thing to really be concerned about as we get to these smaller particles is that if we have typically mobile phases that are really prone to grow things in them. So we want to really be careful that we control our microbiological contamination. These microbes like bacteria can build up in these column beds because of the small particle, basically proliferate and lead to a loss of resolution in our separations. So that's all I really have to talk today about. And with that, I'm going to hand this off to Michelle. Michelle?
Thank you, Stephen. In my section, I'm going to discuss how to use light scattering for size exclusion chromatography on the UHPLC platform. I will start by recapping the advantages of UHP-SEC. UHP-SEC uses columns packed with small particles. As Stephen discussed, it will give much greater resolution in a fraction of time. The peaks are much sharper, so the detection sensitivity is also enhanced. The column dimensions are smaller, both inner diameter and column length, so that will use less samples and less mobile phase. So overall, UHP-SEC productivity is much enhanced. Now, with its better resolution, does it mean we can rely on column calibration to get molecular weight from UHP-SEC? The answer is a clear no. So here is an example of 3 MAP proteins. They should have very similar molecular weights, but somehow they eluted at significantly different elution volumes. If we were using column calibration, we will get wrong molecular weight, especially for MAP3. That's because column calibration assumes the sample of interest has the same conformation and density as the standard used for column calibration. It also assumes there's no column interaction and chromatography is reproducible. In the real world, when one or multiple of those assumptions fail, then we will get very wrong molecular weight. If we not use column calibration but use light scattering, we will get the molecular weight as expected. Here we plotted molecular weight from light scattering against the peaks. And MAP1 has a molecular weight of 146 kilodalton. MAP2 is 152 kilodalton. MAP3 actually has a molecular weight in between, around 150 kilodalton. I will give a quick overview of the 2 light scattering techniques used for online biophysical characterization. The first is static light scattering. Static light scattering is also known as multi-angle light scattering, or MALS. MALS detector measures the scattered intensity in a fraction of a second time scale. And in that time scale, the scattered intensity is a constant, proportional to the product of molecular weight and concentration, based on first principle without making assumptions on the shape or elution properties. It can also potentially measure the radius when the radius is greater than 10 nanometer. But most proteins would have a radius less than 10 nanometer. So to measure the radius and potentially the conformation, we will need another light scattering technique called dynamic light scattering, also known as DLS or QUEL. DLS measures the scattered intensity in a much shorter time scale, nanosecond to microsecond. And that fluctuation as a function of time can reveal diffusion coefficient, and so we can get hydrodynamic radius based on Stokes-Einstein equation on the right. So this is the flow diagram of SEC-MALS, and on the UHPLC platform, we call that micro-SEC-MALS. For ideal system for proteins, the detectors should include UV, MALS with embedded DLS, and a differential refractive index detector, also known as DRI or RI detector. RI detector can measure protein concentration without the knowledge of UV extinction coefficient. And actually, by combining UV and RI data, we can calculate protein extinction coefficient. RI detector is also needed for protein conjugate analysis. By adding MALS and DLS to UHP-SEC, we can now measure the absolute molecular weight in solution without making assumptions on the protein shape or the elution properties. We can also get more information. MALS measures the molecular weight. DLS measures the RH. By combining molecular weight and RH, we can reveal shape information. Light scattering detector is also very sensitive to large species, so they are great tools for detecting, characterizing, and quantifying protein aggregates. They also help identify new peaks from UHP-SEC due to its high resolution. Molecular weight is measured in a fraction of a second, so we can plot all the molecular weight data points across the SEC peak, looking for impurities and signs of on-column dissociation. SEC-MALS is also an important tool to characterize protein conjugates, such as pegylated proteins and glycosylated proteins. When we first use our MALS detector on the UHPLC system, we encounter some challenges. A typical SEC peak volume is above 400 microliter, like in this case, 440 microliter. Our flow cell volume is a little bit less than 70 microliter, so less than 20% of the peak volume, making it acceptable for SEC separation. But when we move to UHP-SEC separation, the peak volume is much reduced to only 1/10. And now the standard MALS detector is no longer suitable for the SEC separation. So if we still use those standard detectors, we will see as the peak moves from UV to light scattering to RI, the peak gets broader and broader. The fragment seen by the UV is now completely lost in the RI trace. It is relatively easy to develop a new dispersion RI detector, and that's what we did first. It's called Optilab UTRex. But it is much harder to make a low dispersion MALS detector. Our R&D scientists and engineers spent the next 2 years develop a new detector called MicroDom. It has a flow cell with 10 times reduction in the flow cell volume and 20 times reduction in the broadening. It also has new laser module and optical bench as well as MALS detector to minimize the stray light to maximize the sensitivity. It also supports dynamic light scattering for simultaneous RH measurement. It has a integrated in situ automatic flow cell cleaning device called Comet. With all those features, we're now ready to use the detector for UHPLC application. Here is an example of a protein sample, IgG, with a fragment. And we compare the results from the two sets of detectors. So here is the standard MALS and DRI detector. The resolution between those two peaks is very poor, and the molecular weights are not correct. And when we use MicroDom and UTRex, then the resolution is recovered, and the molecular weights across peaks are reasonable now. And here is the contrast of the results from those two sets of detectors. Here are the typical experimental conditions we used for UHPLC combined with MALS detection. We used the water's acuity, HPLC or UPLC chromatograph. We typically use the BH200 columns and TUV UV detector. The MicroDom is a MALS detector. Optilab TRex is the DRI detector for the UHPLC setup. Remember those three maps, that they have very similar molecular weights, but eluted at different volumes. We can now use the embedded DLS to measure their hydrodynamic radii. The DLS embedded in the MicroDom has very high sensitivity, can measure MAP down to 20 micrograms per milliliter. And we find all those three maps actually have very similar radii. So this similar radii ruled out the possibility of different elution volumes due to different hydrodynamic volumes. So this would suggest that the different elution volumes is most likely due to different degrees of column interaction. We find the molecular weight from column calibration on the UHPLC platform can give actually more wrong answers comparing to the SEC platform, probably due to its very steep calibration curves. We use the setup to study different stressed IgGs. Here is an example. So we see the monomer peak as the major peak, and there are three main fragments. So the MicroDom and TRex revealed molecular weights and extinction coefficients for those fragments, which help us to identify the different types of the fragments. And as we move to the early elution volumes, we can see the different aggregates, including a large aggregate particle peak and some other partial aggregate forms. We can assign molecular weight to all those peaks. We also studied different loadings. We injected from 0.4 microgram to 6.4 microgram. And as you can see, the molecular weights across those peaks are very consistent. And those molecular weights are also tabulated in this table. The relative standard deviation for both molecular weights are less than 1%, confirming the system is very sensitive and reproducible. MicroDom is an important tool for characterizing protein conjugates. So we use the MicroDom UV and RI to characterize a glycoprotein from the surface of virus on a UHPLC system. And the three detector signals can be used to calculate the total molecular weight and also the molecular weight from proteins and glycans. And so now we can confirm the main peak is the monomer, and there are also dimer and trimer present in the sample. SEC is often used for characterizing the fractions from a prep scale HPLC, as shown here, fractions 1, 2, and 3. But SEC separation time is often more than 15 minutes, and making it only suitable as an offline method. Now with UHP-SEC, the separation time can be greatly reduced. Here we were able to separate all those different fractions within 3 minutes, making it possible as an add-line method for process monitoring. The addition of the light scattering detector can help quantify those different aggregates and fragments. So in summary, by using µSEC-MALS, we can determine the absolute molecular weight, hydrodynamic radius, and extinction coefficient. We can also assess the heterogeneity of molecular weight across the peak, looking for signs for impurity and on-column dissociation. We can use the µSEC-MALS for protein conjugate analysis. And by actually getting more information from those light scattering detectors, we can transfer methods from one platform to the other much easier. The additional information also offers stronger biophysical evidence for biosimilar characterization. The much reduced analysis time may also open doors for new applications, such as the potential method for process monitoring. I will also encourage you to visit our website to learn more about light scattering theories and the applications. This will be the end of my section. I will now hand it to Mark.
Thank you, Michelle. I'm going to go through some case studies with the MicroDom for method characterization challenges. First, I wanted to acknowledge, the group of people within Merck that have helped, with this collaboration. There's been many people across the globe, in California, West Point, and the Netherlands that have helped, incorporate the MicroDom to our workflow. Now we have 6 MicroDoms across 4 sites. In addition, collaboration with Wyatt has helped this project proceed. So first, the biotherapeutics are very complex molecules and require many different assays to fully characterize them. Charge heterogeneity needs to be assessed by IEX. Peptide mapping can do sequence coverage. Today we're going to focus on aggregation and fragmentation, where we look at size exclusion, I think as Steve mentioned, and Michelle, where it was briefly discussed, and how you can use light scattering to help aid in that separation. Application of SEC-MALS, as mentioned, can really help to assess the distribution in your peaks without collection of them, and you can really do it online during separation. You can look at the polydispersity of the molecular weight across a peak to see if there are multiple forms under one peak in the dimer and monomer, and it can really help identify fragments and aggregates. So typically 5-micron columns allow for separation in 30 minutes and provide adequate separation between HMW and monomer. The invention of the 1.7 BEH column allows us to shrink that separation down to about 5 minutes, allowing a 6-fold increase in throughput. We can get equal or better resolution for many MABs, while the reproducibility has been comparable to HPLC. So here we show on the left-hand side an image of the Dawn Helios with the OptiLab TRex refractive index detector. This has traditionally been used for many of our HPLC workflows. It's an 18-angle detector with a larger flow cell. On the right is the MicroDom and the OptiLab Micro TRex instrumentation. We typically use the Acuity UPLC to run this. On the bottom is a comparison of the flow cells for the instruments compared to a quarter. So the large Helios and Trios flow cell has a large volume with the flow path running across the flow cell. The pre-commercial MicroDom also was a smaller version of this flow cell with the flow path running across. They were able to shrink down the volume even more by changing the flow path from going to the top to the bottom of the flow cell and really decrease the volume. So if you look at the table, the Trios total cell volume is 67 microliters, while the MicroDom shrunk down to 4. And this kind of goes along with where Steve was mentioning the importance of system volume and decreasing the band broadening. As you get your columns to be smaller and smaller, for example, 4.6 by 150 millimeter column, the 2.5 milliliter column, that can be substantial. So the first step to bridge HP-MALS to UP-MALS was looking at BSA. And here we show the comparable light scattering profiles for BSA under HPLC conditions and UP-SEC conditions. For the monomer and dimer, we get very good agreement between the HP-SEC and UP-SEC analysis with less than 3%. For the BSA trimer, we get very good agreement between the two numbers, but it's slightly off from the theoretical of about 180. This is likely due to the low abundance in that sample. So the next thing we wanted to look at were multiple MABs and stress MABs, because this is likely to be around on stability to characterize our proteins. And again, HPLC and UP-SEC results are very comparable in the traces. The results for the monomer and dimer are in very good agreement for stress MABs and on-stress samples. So we feel very comfortable that we can bridge the UP-MALS and make it our platform. So in doing some of this investigation, we found one of the MABs was sensitive to the aggregates being not present in PBS. So if you run your sample in PBS on the top chromatogram, we weren't seeing many aggregates. We did some optimization. If you look in the literature, there are different types of modifiers that can be used, such as organics. Sulfate is a common modifier. Here we found a modifier that was capable of improving the recovery and separation. So we can use the MicroDom and light scattering device instrument to really help us answer, where are the aggregates? Are they stuck on the column, or are they co-eluted with the monomer? So we first looked at the molecular weight of the monomer in our optimized mobile phase, and we're given about 139 kilodaltons, which is much less than we would expect. Most MABs typically we see 150 kilodaltons. So we looked at our BSA, and we found that was coming out much lower than expected, 61.7, which is outside the accuracy of the light scattering measurement, which is usually 5%. So the fundamental question we had is this modifier changing our dN over dC for our proteins when we use the refractive index detector to calculate the concentration? So for many proteins in PBS, the dN over dC is 0.185. So we wanted to take a look into that a little bit further. Understanding the importance of dN over dC in measuring the molar mass, in addition to using it as your concentration source, in the light scattering term, the dN over dC is squared, even if you're using UV as your concentration source. So any error in your dN over dC, a 10% error, would result in a 20% error in your molar mass. I think this is one of the things that I found very interesting, is that even when you're using UV, the dN over dC is still critical. And it may be important, it may just be a reason to use the refractive index as your concentration source. So the UT-REX flow cell design allows for absolute refractive index measurements and differential refractive index measurements. The absolute refractive index measurements are made when the mobile phase and analyte travel through both the sample chamber and the reference chamber, and the refractive index can be measured. The differential refractive index can be measured when mobile phase is in the reference chamber and analyte and mobile phase pass through the sample chamber. So the first thing we wanted to do was take a batch mode measurement of the dN over dC, where you can prepare 5 or 6 different concentrations and inject them directly into the refractive index detector and look at the DRI. You can then fit a curve to that and calculate your dN over dC. Here we inject mobile phase as our controls, just to see the refractive index due to the mobile phase as it comes into contact with the mobile phase that's running. So here we got a calculated dN over dC of 0.162. So we kind of found that the results were not reproducible when we repeated this. This is likely due to the accurate concentrations that are required for this measurement. In addition, buffer exchange is necessary to remove excipients from your product. And in fact, Wyatt recommends drying your protein and then reconstituting it in a liquid to get accurate measurements. The second method that we can use to calculate the dN over dC is published in this paper, and it's also recommended from Wyatt, where if you know the dN over dC in solvent A, and you know the refractive index of solvent A, or you can measure it, you can measure the refractive index of solvent B and correct for it. So here we measured the refractive index of PBS and our solvent B. And using the partial specific volume of 1, we come up with a dN over dC of 0.171. So we reprocessed our BSA and MAB3 using the dN over dC of 0.171. And now we see an expected, the molecular weight of BSA is 66.8, which is very close to the expected molecular weight. For our MAB3, we see very close to the expected molecular weight, and now our HMW is corresponding to that of the dimer. So the next question we had is, can this dN over dC be applied to other MABs as a platform? So using our platform UHP-SEC-MALS with this modified mobile phase, we see good agreement between the monomer in PBS and the monomer in the modified solution for 6 different MABs. So it was very good confidence that this can be used as a platform, being that we're only correcting for differences in refractive index in the 2 mobile phases. So we've successfully transitioned to the UHP-SEC-MALS with modified mobile PBS. So the question we were trying to answer is, where are the aggregates in MAB3? And the top chromatogram shows PBS, a light scattering, an overlay of the light scattering in UV traces. We can use light scattering due to its increased sensitivity for higher molecular weight species. You can kind of see a little blip in the light scattering trace there in PBS, but we don't really see anything else. When you overlay the same light scattering trace in UV in our modified solution, you can really see the aggregates there and the added sensitivity from the light scattering detector. So based on this, we were able to kind of feel that the aggregates were stuck on the column and our modifiers suppressing those non-specific interactions. I think this is one of the challenges Steve mentioned during his talk, is the separation needs to be based solely on size, and we have to add modifiers to counteract those non-specific interactions. So we can also use the MALS for method troubleshooting during method implementation. We had MAB3 as our reference standard throughout multiple runs, and an analyst identified this little shoulder that they never saw before on a brand new column. So the first thing they thought was, oh, my column's bad and I need to throw it out or discard it. Looking at the separation, we can see there's very good separation between the dimer and the monomer. So it was unlikely that the column was bad. So the question was it a fragment or a sample artifact, or was it some sort of carryover? I think, as mentioned earlier, you can use 2 columns to increase your separation capabilities. So we hooked up 2 columns in series with a very small tubing to decrease band broadening, and you can really improve the separation between the monomer and those low molecular weight fragments. We also tested a stress drug substance to see if this peak grew on stability, and you could see an increase in the aggregates as well as fragmentation. So we've identified that the peak is real, and it is stability indicating, and we would have to deal with it during stability testing. Is it a fragment or some sort of conformational isomer that can form? We can use the MicroDom to help answer that question. So the top shows the molecular weight distributions of the drug substance and stress drug substance. You can see for the monomer and dimer peaks, the molecular weights overlay very nicely. The red represents a stress drug substance, and now you can detect this 93 kilodalton fragment, and we come up with a molecular weight for 35 for the second fragment. This is the UV trace for the corresponding sample from the previous slide. Looking at the Yang et al. Paper, we saw some hinge region fragmentation that has a 100 kilodalton fragment, and that kind of coincides with what we see for fragment number 1, 93 kilodaltons. Fragment number 2 would be the corresponding 50 kilodalton fragment. We are measuring a 35 kilodalton fragment. This is likely due to the low abundance of that peak. As you go down in molecular weight, you need a lot more to get accuracy in the light scattering. So as that peak grows on stability, we may be able to get much more accurate molecular weight data. So the MicroDom can allow identification of peaks where you would normally need to isolate and characterize them through mass spec, and it can give us confidence in what we're seeing. So in summary, the UHP-SEC-MALS and HPSEC-MALS show comparable performance, and we were able to successfully transition to our platform. The mobile phase conditions can impact DN over DC, so when you're adding exotic modifiers, it may be worthwhile to double check the DN over DC that you're using. The UHP-SEC with the MicroDom allows for faster analysis time. If you're using the UHP-SEC for release, then you can directly measure and characterize the peaks without bridging. The MALS can help aid in SEC method characterization when challenges arise during implementation. And lastly, it can focus collaboration with why it is critical, especially your local support. They're always there to help and are very quick to answer. So I think that's all I have. And any questions?
All right, thank you, Mark, and we can move on to the Q&A session now. The first question is, what role does the particle size distribution play in the SEC resolution and band broadening, and how do you control it during manufacturing?
I'm going to guess that question is for me. In terms of the packing efficiency of the column, having a reasonably good control over the particle size distribution is important. You're going to get more efficiently packed columns in terms of just plate number as the particle size distribution kind of reaches an optimum, and that's really kind of determined empirically where that needs to be. What was the second part of the question?
How do you control it during manufacturing?
Particles are generally manufactured and then sized, and a cut of a specific size range is taken for that.
All right, the next question, can we use intrinsic viscosity, an intrinsic viscosity detector to calculate RG instead of DLS?
I guess that question would be for me. Yes, we can also use online viscometer to measure the intrinsic viscosity. Then based on that and molecular weight, RH can also be calculated. So we actually have demonstrated of using the online viscometer to get the RH for proteins, and often it can be more precise comparing to online DLS. But I don't think there's a suitable viscometer for UHP-SEC separation. But for the regular SEC, yes, we can use an online viscometer.
What typical DN over DC values are used, and what consideration should we take to select the proper DN over DC?
So I guess I can take that question. Typically, we use 0.185 for PBS solutions. I think in terms of consideration, should we take, I think viscosity impacts the DN over DC slightly. And when you're measuring the DN over DC, in batch mode, like as we did in method number 1, the concentration range that you pick to measure those has to be within tenfold of your expected DN over DC. So I think it's recommended to use usually 0.1 to 2 mg per mil, and that concentration range can really impact the DN over DC.
How is the lot-to-lot column repeatability of those BEH-SEC columns? What is your QC control, and is it the relative standard deviation of the protein standard retention time?
So that's going to be my question. So in terms of lot-to-lot variability, Waters does manufacture everything from the ground up for these particles. So we control the manufacture of the particles as well as then the pore enlarging of the particles all the way through to the modification of the surface. The QC tests are included in the C of A, and the specifications that those run under are also included in there, and it really kind of relates to, we look at a number of different tests, including a lot of tests that aren't on the C of A, and those are more or less proprietary, and I can't really talk about those. But we also include a regular standard protein test that's included on the C of A for the each batch of material. For each column is manufactured or packed to a target efficiency, and that efficiency is evaluated on a column-per-column basis. And it has to meet a specification for that.
The next question is, I did a SEC-MALS analysis on a product, and there is no signal in DRI or UV, but a sharp peak in light scattering at large molar mass. How should I interpret the light scattering result? Is it a real quick protein? Is it a real protein peak or noise signal from the column shedding?
I can take that question. Excuse me. We can first make a blank injection. Usually it's an injection of the mobile phase, and make sure the peak is not coming from the column. So if it's a column shedding, due to column shedding, then you will see that in the blank injection. If you don't see the same peak in the blank injection, then you know that's part of the sample. And actually, based on light scattering detector alone, we can also measure the size of that peak, even count the number of particles.
Is the Dawn Helios 2 incompatible with UHP-SEC-MALS?
The Helios 2 is not ideal for the UHP-SEC. If you use Helios 2, you may use the UV as the concentration detector. But if you try to also use the downstream DRI detector, then the dispersion will be too excessive, and the resolution that you worked hard from the column can be lost. So it's best to use the MicroDom with TRex for the UHPLC system.
What are the principal differences in mechanism of separation of UHP-SEC and regular SEC?
Mark, do you want to take that, or would you like me
Sure.
to?
Sure, I can take that, Steve. I think the principal differences are very similar. I think you have particles traveling through a porous bed. I think one of the advantages that we're taking advantage of are the smaller particle sizes. Allows more packing material to get in there. So in terms of fundamental principle is the same. You just have more particles, more packing material in a small column, so you can go to much higher flow rates and really maintain separation. There is a question of always the high pressures inducing aggregation, but it's pretty well documented from most of the MABs and some therapeutic proteins that hasn't been the case, and I haven't seen or read of any antibodies having that. And Steve, you can feel free to add to that.
Well, thanks, Mark. I think that's pretty much spot on. And just remember the Van Diemter curve. The smaller the particles, the faster the throughput. I sound like a marketing person.
Does UHP-SEC cause more shear degradation than SEC?
I think I could take that. From our experience in running comparative runs for different proteins, the shear forces are not that significantly different between the two separations, giving the changes in linear velocity that we run across of these columns. So if we do side-by-side analysis, and obviously this can change depending on the size of your protein, fragility of the protein, conditions you're running, but if we compare lower flow rates to higher flow rates, we usually do not see a difference in terms of the amount of fragmentation that we see. And that's really the easy test to tell whether or not that's happening.
All right, that's all the time we have. Thank you again, Stephen, Michelle, and Mark, for your fascinating presentation, and thank you, participants, for being a great audience. Be sure to check CNEN or CNEN Online for information on the next edition of CNEN webinars. Thank you to On24 for technology and production services, and thank you Wyatt Technology for the sponsorship that made this interactive webcast possible. For CNEN webinars, I'm Alexander Taylor. Goodbye.